Hello, everyone, and welcome to my presentation on the investigation into the role of microbiota in the development of head and neck squamous cell carcinoma. This research was conducted at the Texas A&M School of Engineering Medicine under the guidance of Dr. Kamlesh Yadav and Dr. Shamir Kader. To provide some background on this topic, head and neck squamous cell carcinoma is a cancer of the squamous cell membrane that lines the nasal, oral, pharyngeal, and laryngeal cavities. One interesting feature of this cancer is that in some instances, it can be caused by the human papillomavirus, or HPV. This helps us divide the broader topic of head and neck squamous cell carcinoma into two main groups: HPV positive and HPV negative. One study published in 2017 showed that both of these strains showed the presence of 3 lactobacillus bacteria in the saliva of patients: Lactobacillus gasseri, Lactobacillus johnsonii, and Lactobacillus vaginalis. These bacteria are typically responsible for the healthy regulation of the vaginal microbiome, so their presence in both strains of head and neck squamous cell carcinoma is of interest. The objective of this study was to explore potential connections between the metabolic pathways of these lactobacillus derivatives and the pathogenesis of head and neck squamous cell carcinoma. This was done in 3 steps. The first step was to identify a list of genes that contribute to the tumor pathogenesis and conduct a gene pathway analysis with Enricher to specify pathways implicated in this process. Then, we conducted a comparative analysis with the biopsych database to identify overlapping pathways between humans and the 3 bacteria in question. Lastly, these 2 lists were compared against each other to identify pathways found in common and provide a potential connection. The common pathways that are shared by the microbiota and the disease pathogenesis included altered amino acid metabolism, several components of the glycolysis pathway, and portions of the DNA mismatch repair pathway. Lactobacillus gasseri was involved in biochemical modification of macromolecules, including post-translational modification of lipoproteins. Lactobacillus vaginalis was identified to be involved in amino acid metabolism, biosynthesis of carrier proteins, and the degradation of carbohydrates. Lastly, Lactobacillus johnsonii was involved in the degradation of nucleotides and nucleosides. The table in the middle of the poster shows the numbers of shared pathways between human tissue and the 3 bacteria generated by the biopsych database. In parentheses, the Jacquard similar coefficient is also provided. The figure below shows the results of the Enricher analysis depicted in increasing p-values. These p-values were derived from the KEGG 2021 Human Pathways Database. In conclusion, there were some pathways identified that were overlapping and others identified that were not. While not all these pathways are directly correlated with the pathogenesis of head and neck squamous cell carcinoma, their functions can be appreciated in the proliferation of tumor cells. The presence of these lactobacillus derivatives in the saliva in these patients suggests a connection in the altered tumor metabolism. Further studies should be conducted to isolate the specific pathways that contribute to the progression of head and neck squamous cell carcinoma. Lastly, at the bottom right, you can find my references for this project along with my acknowledgments of the wonderful advisors at Texas A&M with the Engineering Medicine program. Please feel free to contact me at the provided email with questions. Thank you for attending my presentation.